Hi everyone, this is Arul Jothi from the Department of Biochemistry. In this video, we are going to see about the cell cycle and cell division. Cell cycle refers to the series of events that takes place in a cell resulting in the duplication of DNA and division of cytoplasm and organelles to produce two daughter cells. The phases of cell cycle, it includes the maturity and subsequent division. These events include duplication of its genome and synthesis of cell organelles. Human cells exhibit a typical eukaryotic cell cycle. It takes around 24 hours to complete one cycle of growth and division. But the duration varies from organism to organism and even cell to cell. The first phase is the interphase. Actually, interphase is the resting phase of the cell cycle. The time during which the cell prepares for division by undergoing both cell growth and DNA replication. It occupies around 95% of time and all over the cycle. The interphase further divides into G1 phase, that is gap 1 phase. It happens between the mitosis and the initiation of replication. Then S phase, that is the synthesis phase. DNA replication will take place during this phase, and G2 phase, the gap 2 phase. During this phase, the RNA, proteins and other macromolecules required for multiplication of cell organelles and spindle formation and cell growth are produced as the cell prepares to go into the mitotic phase. Then M phase. In this phase, mitotic phase or the phase of equational divisions happens. The cell undergoes a complete recognition to give birth to a progeny. It includes four stages, that is prophase, metaphase, anaphase, and telophase. The next one is the cytokinesis. In this phase, the cytoplasm of cell divides. It begins as soon as the mitosis ends. Plant cells are much tougher than the animal cells since they have the cell wall. Thus, cytokinesis occurs in plant and animal cells in a different manner. Mitosis, it is the process by which a eukaryotic cell divides to produce two identical daughter cells. The process occurs in the somatic cells and ensures that each daughter cell receives an exact copy of the parent cell's DNA. The mitosis consists of four stages. When the prometaphase is there, it happens as a five stage, that is prophase. Chromatin condenses into a visible chromosome. The nuclear membrane begins to break down and the centrosomes move to the opposite poles of the cell and the spindle fibers start forming from the centrosomes. In the prometaphase, the nuclear envelope completely disintegrates and spindle fibers attach to the kinetochores of the chromosomes. Then metaphase, where the chromosomes align at the metaphase plate, that is an imaginary line in the center of the cell. The spindle fibers are fully attached to the kinetochores, ensuring equal separation of chromosomes. And in anaphase, the sister chromatids separate as spindle fibers pull them to the opposite poles of the cell. The cell elongates as non-kinetochore microtubules push against each other. The fifth one is the telophase. In this phase, the separated chromosomes reach the poles of the cell and the new nuclear envelope forms around each set of chromosomes. Thus, the chromosomes decondense back into the chromatin. The spindle fibers disappears in this stage. Then coming to meiosis, meiosis is a special type of cell division that occurs in germ cells, that is um, sperm and egg cells to produce haploid gametes. Unlike mitosis, which creates two identical daughter cells, meiosis results in the four genetically unique daughter cells. The phases of meiosis includes meiosis 1 and meiosis 2. In meiosis 1, uh, the the chromosomes number is reduced by half, that is reductional division, and in meiosis 2 it is an equational division like mitosis. In under meiosis 1, it includes prophase 1. Prophase 1 is the longest and most complex stage. In this stage, the chromatin condenses, homologous chromosomes pair up to the tetrads, and crossing over occurs. In prophase 1, there are five substages, that is leptotin, zygotin, paketin, diplotin, and diakinesis. Then the second stage is the metaphase one, where the tetrads align along the metaphase plate, spindle fibers attached to the homologous chromosomes. Then anaphase 1, homologous chromosomes are pulled apart towards the opposite poles and sister chromatin remains together, unlike in mitosis. Yeah, in telophase 1, the chromosomes reach the poles and a new nuclear membrane forms temporarily. Cytokinesis occurs, dividing the cytoplasm and forming the two haploid daughter cells. In meiosis 2, the equational division, that is similar to that of mitosis, will take place. Prophase 2, chromosomes condense again and spindle fibers reform. Nuclear membrane dissolves. It was if it was reformed after meiosis 1. Then in metaphase 2, chromosomes align at the metaphase plate. Spindle fibers attach to both sister chromatids. Then anaphase 2, sister chromatids separates and move to opposite poles. Telophase 2, chromosomes decondense and nuclear envelopes reform around each set of chromosomes. Cytokinesis occurs, resulting in four genetically unique haploid cells. Thank you.